The overall goal of this imaging method is to study subcellular organelles in a vertebrate in vivo.
This method can help to answer key questions in the field of neuronal cell biology, such as dynamics of mitochondria or centrosomes in an in vivo context.
The main advantage of this technique is that the entire experiment can be performed in an intact vertebrate without the need for any surgical procedure.
This method can provide insight not only into subcellular dynamics under physiological conditions, but it can also be applied to studies of disease models, such as Alzheimer's disease.
After generating transiently expressing embryos by microinjection, or obtaining embryos from stable transgenic lines according to the text protocol, at the end of day 1, screen for dead or unfertilized eggs. Using a plastic transfer pipette, transfer the viable embryos to Danuse solution containing 1-xPTU to prevent pigment formation, and maintain the embryos in Danuse solution containing 1-xPTU until screening for transgene expression. On the day of the imaging experiment, use a fluorescence dissecting microscope to screen and anesthetize embryos for transgene expression. Then transfer the selected embryos to a separate petri dish containing Danuse buffer with 1-xPTU and 1-xtricane. Once the embryos have been anesthetized, gently pipette a few into the low-melting agarose containing 1-xPTU and 1-xtricane, transferring as little liquid as possible to avoid diluting the agarose. Transfer the embryos with a small amount of